Good afternoon. My name is Emily Finn, and I'm a project coordinator in the Center for Human Brain Variation. I'm very excited to welcome you all to our center's first seminar series. One of our center's core goals is to increase accessibility to neuroscience research and foster a collaborative scientific community. To help reach this goal, we created this seminar series. Our seminar series is open to the public, held in a hybrid format, and the presentations are designed to be accessible to a wide audience. Today, I'm honored to introduce our inaugural speaker, Dr. Sabina Berretta. Dr. Berretta is an associate professor of psychiatry at Harvard Medical School, the director of the Harvard Brain Tissue Resource Center, director of the Translational Neuroscience Lab at McLean Hospital, and a Broad Institute associate member. Dr. Berretta aims to promote rigorous research on nervous system disorders. Her work includes overseeing and expanding collection and distribution of brain specimens and developing novel approaches and methodologies to do so. She is committed to combating stigma associated with brain disorders. The more we learn about the biology underlying brain disorders, the more effectively we can correct misconceptions and to reduce stigma. The Center for Human Brain Variation and the McCarroll and McCoskell labs work closely with Sabina to characterize variation and abnormalities in the human brain of hundreds of people. Dr. Berretta's remarkable work as the director of Harvard's Brain Tissue Resource Center is what enables all of us to conduct our research and expand our understanding of the human brain. Please join me in welcoming Dr. Berretta here today to discuss the role of postmortem human brain studies in research on nervous system disorders.
Thank you for a wonderful introduction and for inviting me today. I'm really delighted to be here, and thank you for choosing to spend a Friday afternoon listening to my talk. So we are going to talk about working with human postmortem tissue and investigations that we can do using this system. What is important to understand is that the brain itself and brain disorders are so profoundly complex that we really need to use all the tools we have and invent some more in order to really get to understand both how our brain works, but also importantly, what happens when the brain changes and we have a brain disorder. So this is meant to really represent all the approaches, each with its pros and cons. We need to be able to use and kind of integrate together in order to do this work. But going to the topic of today, I should mention that the Latin part of the title of my talk, Mortui, Vivos, Docent, is a little joke of myself. I hated studying Latin when I was in school, and I was so bad that I almost didn't make it into medical school. So this is meant as a joke on me to include Latin. My professors will be really laughing. But more seriously, the dead teach the living was the motto that anatomists in the Renaissance around 1500, 1600 used to explain why they need to work on cadavers, why they needed to dissect human cadavers in order to understand the human body. And indeed, we have a lot to learn from the dead, in our case, from brain donors. First, because we know very little about the brain, particularly the human brain, and I'll explain why specifically the human brain is something that it's important to focus on. But also we have to learn from the generosity of brain donors and all their families that choose to use their death to give us a gift of knowledge so that we can do this work. So I'd like to acknowledge them and express gratitude for all of them, for all the thousands of brain donors that make our work every day possible. Before going ahead, I would like to also thank another group of people, and this is my colleagues at the Brain Bank and my lab that also work in part at the Brain Bank. Without their dedication, compassion, professionality, I wouldn't be able to be here telling you all about this work. So I hope they will forgive me for asking them to stand up so that I can introduce them. So before you sit, Isabel Darling, a tissue coordinator and donation coordinator. Kylie, a researcher in my lab, but also donation coordination and tissue coordination. Lizzie is Elizabeth Lawton, a researcher in my lab, but also a tissue coordinator and dissectionist. Phyllis Sodur, a tissue coordinator. She's the one, together with others and Isabel, that prepared tissue for every investigator that requested. Catalina Tabit, a clinical liaison, so she coordinates brain donations together with others that are not yet here or may not be able to come. And Josh Abston, who is a data analyst in our lab, but also a tissue coordinator and a dissectionist. So thank you, all of you. So going back to the dead teach the living, this motto started at the time in which Vesalius started his work in the 1500s. He is considered to be the father of modern anatomy. Vesalius was originally from Belgium, but went to work and teach anatomy and medicine at the University of Pavia in Italy, which together with Padova and Bologna were really the center of the medical learning at that time in Europe. Vesalius realized that the knowledge that had been taken as unchallenged as a dogma for the past 1500 years was actually based on dissections on other species, and therefore had a lot of mistakes. So it was work that had been taught initially by Galen, and for over thousands of years, 1500 years, it went completely unchallenged. Vesalius is known to have found at least 200 major errors in the work that Galen described, because Galen couldn't dissect human bodies, was basing it on other species. So for instance, famously, the description of the pulmonary blood circulation, the Galen had expected the heart ventricles to be able to communicate with each other, which in fact they don't, but nobody ever challenged it. And Vesalius and eventually Miguel Servet, one of his students, demonstrated that that wasn't the case. So Vesalius described the full human anatomy, brain included. He's the first one who had made, among many other things, a mold of the brain ventricles, so to understand better how they work. He published his famous work, De Humani Corporis Fabrica Libri Septem. Libri Septem is 7 books of human anatomy. And he was able to do it because dissecting cadavers was forbidden under the Roman Inquisition, but he had connections with magistrates that looked favorably at his work, and therefore allowed him and eventually his disciples to do it. So I bring up this example to say that it is really important that we use animal studies and animal models, but also integrate them with knowledge that we need to have on the human body and the human brain in this case. I would like to remember Camillo Golgi as studying many different species and inventing the Golgi impregnated neurons that allow him and eventually Ramon y Cajal to describe the cytoarchitectonics of the brain in several different species, including human. As you might remember, Camillo Golgi and Ramon y Cajal won together the Nobel Prize, although they were arch enemies and they had completely different theories on how the brain actually is structured. The funny part, if you notice, I can point at it, but Camillo Golgi used an actual razor to cut sections for his Golgi impregnation. So even before our microtomes, he was simply using a razor from home. So why study the human brain? There are obvious species-specific differences. both obviously in size, but also in cytoarchitectonics. Jenny, Edward, and Karen, Joseph are also part of the brain bank. They are clinical liaisons. I just want to introduce them as well. There are major differences that are not only in size, but they are obviously in cytoarchitecture of the brain. It doesn't necessarily mean that our brain is better than any other species. If I were to compete with a mouse navigating a dark space that I don't know, the mouse would win and would think I'm an idiot. But there are differences that depend on adaptation during evolutions. So it is important that we understand them well. And much work, including work done by Steve and Fennakrenen, show that it's not only at the level of gross morphology and the cytoarchitectonics that the different species are different, but that in fact there are adaptive changes across evolution that can affect the abundance of different cells, change the genetic program within a cell type, allocate different locations or resources, or even in some cases invent a new cell type. And in this work, I won't go in much detail, but they took 4 different types of interneurons with standard markers that we use to recognize them in 5 different species, and then picked genes that were uniquely or predominantly expressed in each of these cell types, showing that in fact that these are not conserved, and the markers that we would have used to look at those cells across species in fact may not be as valuable as we thought. And obviously, needless to say, we need to pay attention to pathology. Looking at the human brain is the only way to really understand changes in the brain at the level of molecules and cells in any pathology. And I'm making very obvious examples here, for instance, Alzheimer's disease with the classic neurofibrillary tangles and neuritic plaques. Very standard, very well-known pathology. We probably know very little about what else is going on and what precedes or even causes some of the symptoms in Alzheimer's disease. The only way to look at it, to understand it, is through the human brain. Same with Parkinson. The bottom is unaffected, and you can see, you can tell here because of the, you can see the substantia nigra pars compacta with plenty of dopaminergic neurons in it, while above is the brain of somebody that had Parkinson's disease, and you can see that almost all dopaminergic neurons are lost. How do we know this? We need to look at the human brain. In psychiatric disorders, we typically don't see these very important changes, but this was one in a lifetime in which Harry Pantazopoulos, who was in the lab, now he has his own position at the University of Mississippi, found changes that we could literally see by holding the slide to the light. These are changes in extracellular matrix, which we published quite a long time ago. They were so striking for a disease such as schizophrenia that when I showed this photo to our program officer at NIH, he told me, you took a Sharpie and made those marks yourself. Eventually he came around. So how can we study the human brain? We need brain repositories. Without collecting human brains and being able to study them and be able to distribute them out, we wouldn't be able to do these studies. So I'd like to tell you a little bit about our brain repository, the Harvard Brain Tissue Resource Center. The first thing important is, what are we trying to do? We're trying to advance research on the human brain and of course on brain disorders. And how do we do it? We collect brain donations from across the country, and we redistribute, we characterize each donor painstakingly, and we redistribute tissue out to investigators across the world. So I'll tell you a little bit about how that happens. We are part of the NeuroBioBank. This was founded in 2013 as a way for NIH to put together several different brain repositories that now work as a unit with harmonized protocols and collaborate on brain distribution, on sample distribution. And this is particularly important because investigators don't need to go brain bank by brain bank asking for samples, but can request tissue directly through the NeuroBioBank and have it put together by several different repositories. And you can see there which one they are. We can accept brain donation from anywhere in the country, and this is one of the strengths that our tissue repository, together with another 3 part of the NeuroBioBank, has. And then we redistribute thousands of samples every year. This is over 10 years, not only within the United States, but also across the world. And therefore, we are able to make research possible really for many, many different laboratories. And this is a shameless plug. Everybody can sign up to be a brain donor. It really helps research. So very briefly, how does it actually work? It seems such a kind of exotic thing to do if you don't actually know how it works. So the first thing to know is that timing is absolutely important. From the time a donor passes to the time the brain arrives to our laboratories and we dissect it, we have less than 24 hours. And this can be if the donor was around the corner or if they were on the other side of the country or in the middle of nowhere. So what we need to do is work very closely with the families. They need to notify us either immediately after death or if possible when death is imminent, so that we can start organizing the really complex orchestration of a brain donation. So the first thing that people need to do is call us. We have a brief screening. After we screen and we can accept them, we can process a informed consent in a secure manner. Once we have a consent, we have a network of technicians and pathologists across the country that work independently, but that we can call to go and recover the brain. So we need to find the closest to the donor. We also need to find where the brain recovery can happen, which sometimes is a challenge. It is transported to our laboratories, which are at McLean Hospital, either putting them on a plane and then, of course, driving them over or driving them in some cases. And we dissect the brain in a way that I will show you. Fair warning, I'm going to show you photos of fresh human brain. It's good to know what it looks like as you will be working on it or might be working on it. So we take photographic records across the dissection. Some of the people in the room just dissected these 2 brains this morning, so they have these images fresh in their mind. Once we take photographic record and we weight it, we separate the 2 hemispheres. One hemisphere is put in formalin and will go to neuropathology. The other hemisphere is dissected. So we first remove the cerebellum and the brainstem. We dissect the cerebellum sagittally. We dissect the brainstem coronally with respect to its main axis, which has some implications that I'm not going to go through, but I'm happy to discuss. And then we dissect the whole hemisphere from anterior to posterior in 5 millimeter sections. It's photographed and then it's frozen in liquid nitrogen vapors. And then they are stored, the frozen component, in -80 freezers. We pick them up again whenever we need to distribute them. In the meantime, we need to characterize the donors, each donor, the best we can. Because when we accept a brain donation, we have very little time to learn about the donor. We need to do things very, very quickly. So we work collaboratively with the families after the brain donation to retrieve health records and a family questionnaire. And in the meantime, we also do a full QC analysis, and including serology, toxicology, and a neuropathology report. So this is our neuropathologist, Derek Oakley, working with one of us. dissecting the brain, the fixed part. And once we have all this information, we do a consensus diagnosis, so sort of like a case conference in which now we have all the information for each donor. We discuss it and we try to figure out with a set of diagnoses. And this is actually a really interesting component of this work. We have the privilege of seeing both the full clinical course of the disease, but also the brain of that person. And the interesting part is that at times the diagnosis in life and the neuropathological diagnosis don't match. And this is work, for instance, that Catalina and Karen here in the audience are developing. So for instance, in this example, we looked at all the donors that came in with the clinical diagnosis of frontotemporal dementia and asked at neuropathology, are they all frontotemporal dementia? It turns out that only 37% were purely frontotemporal dementia. Another 39% had a mixed pathology, so there was frontotemporal dementia together with many other things. And about 23% didn't have FTD at all, well, with the exception of Pick's disease would be technically FTD. But you can see the smattering of other diagnoses that we see. And although I'm not going to discuss this, this is a really interesting point. It doesn't mean that the clinicians didn't know what they were doing. It means something a lot more fundamental, which is that we actually don't know how changes in the brain translate into symptoms. We think we do. We think that we look at Alzheimer's and say, well, they have neurofibrillary tangles or senile plaques, and that's what causes the cognitive decline. But do we really know that? And there are many other symptoms in this example in Alzheimer's that may or may not be accounted for by the classic neuropathology. So that is something that I think our field really needs to pay attention very closely. So once we characterize the brain, we have everything. We can distribute them out. That is the website. And of course, it goes to investigators for their work. The interesting part about the brain donation is something we tell people is a brain donation is a gift that keeps giving, because each brain really goes into hundreds of studies all across the world. We only distribute enough for an investigator to do their work, but no more than that. And therefore, each brain goes into many, many, many different studies. For those that are interested in requesting tissue, I'm happy to discuss it later, but it is done through the NeuroBioBank portal. You can see that you can figure out not only which repository you want the tissue, or many different repositories. You can look for diagnosis, for neuropathology, for many other different characteristics. Some of them are not in there, they are not posted by the NeuroBioBank, and they have varying difficulties for us to extract them from the health records. But this is information that we can share with investigators when needed. When we send tissue, we typically send the neuropathology summary or even the full report if needed in a de-identified manner. When we have clinicians' notes, we share those. Many other things need quite a bit of work for us to extract, but they're possible in order to do a full analysis. So something that I thought could be important to bring up is work that Steve and I and our groups have done in collaboration. A lot of work extracting information, and then Olivia has been doing quite a bit of analysis together with Seva, is how do we predict how well tissue samples will perform? And the reason this is important is because there are a lot of myths and misconceptions on what is important and what we should really look at. And of course, this depends on the assay that we are preparing, that we are planning to do. So in our case, we looked at single nucleus RNA-seq studies, so that what I'm gonna tell you may be most relevant for this type of studies. But the findings, some of the findings were actually particularly surprising. So briefly, we had a fairly large cohort. We had a cohort, and I'll tell you a little bit more about the study with schizophrenia and unaffected controls later. where we had almost 200 donors, 191. Plus, we had unaffected control from other 3 brain regions, caudate, BA4 and BA32, from other studies that we're doing in collaboration. We included donors with schizophrenia after realizing that there wasn't a difference with regard to the quality metrics that we were looking at. It wasn't true for the other donors included in the other studies. So all of you will be familiar with this. The metric that we focused on was UMIs per nucleus, so the number of RNA transcripts or unique molecules that we were able to ascertain per each nucleus as a measure of the RNA or the possibility of RNA degradation in each of the cells. It varies by cell type. I'll show you a little bit of it. So it is important to do it by cell type. We worked, as all these studies have done, in a village, so in batches of 20 donors together. I'll tell you a little bit more later about how it is done. Many of you are already more than familiar. And this is how many nuclei we were able to analyze for BA46, over 1,200,000, and then several others for the other brain regions. And here I'm going to show UMIs per nuclei for only astrocyte, glutamatergic and GABAergic neurons, although we have data for other cell types too. So the first thing that we could see, so these are correlation between across 2 brain regions, caudate and BA32, caudate and BA32 for projection neuron, interneurons, and astrocytes. And you can see that astrocytes seem to be a lot less vulnerable to degradation. Neurons are a lot more delicate, a lot more vulnerable. And this is something that has been seen in many, many different studies. The other interesting part is that brains that underperform in one region also underperform in others. So that in fact, doing work on one brain region in this, with using this assay might tell you how the brain will perform for other brain regions as well. So we looked at many, many different variables, and many people in the room have been kind of digging through health records and try to find variables. But I'm going to show you mostly the one that I'm listing there. The first that could have been a surprise, not to us, because we have been saying PMI, postmortem time interval, does not really give you a very good index of tissue quality. And in fact, it doesn't. It has no predictive value whatsoever. It is true that most of the variants come from about 15% of the brains that underperformed, but PMI did not predict that. Now, PMI, postmortem time interval, is usually calculated, different labs calculate it in different ways. We calculate it in a fairly stringent manner. So from the time of death to the time we're actually done with the last piece of tissue put in a freezer. So it's a fairly long span, but not only didn't have any effect, but you can see that we had PMIs that usually would never be considered that actually perform relatively well. Even at 30 hours, that wasn't a problem. Other factors obviously do have an effect. So which one are they? RIN, RNA integrity number, does robustly correlate with UMIs per nucleus. If you calculate it, if you measure it from the same tissue block, RINs change region by region. So taking it from one brain region does not completely predict the RIN for the whole brain. So it's important to do it from the same tissue block. But the other thing I would like to point out is that even for RINs that are typically considered to be well below what one should use, such as RIN of 4, you have plenty of donors that performed really well. So it's important, but it doesn't tell you the full story. pH did matter, and that's actually an older measure that we used to take all the time, we take regularly. It's taken across the whole brain. It does seem to actually predict quality fairly well, mostly for neurons as you can see, again, not so much for astrocytes. And then we wanted to take a look at autolysis. So this is something that happens to the tissue that typically we detect in the cerebellum, in the granular cell layer, that may reflect several different factors. That may be perimortem or postmortem. But it's typically assessed mostly as not in a very quantitative manner as absent, mild, moderate, and severe. And it was only the moderate and severe that explain some of the performance of the tissue, although not entirely. And just for you to know what is that we are looking at is that's a granular cell nuclei within the cerebellum. And it's difficult to see if I don't go too much in detail, but you see the difference between what is considered to be no autolysis and what is considered to be severe autolysis. Autolysis really means the nucleus of the cell is deteriorating. So it becomes really smudgy and there are a lot of vacuoles. So it is not that surprising that it's telling us something about the tissue. And then we were interested in understanding what we called worm time. It was a bit difficult to extract the information across several different decades of tissue collection. But we calculated it from the time of death to the time the body was placed in refrigeration. And it had some effect, although not as much as we could have expected. So just to summarize this part, most variance in the sample seems to come from about 15% of the donors. We tried to understand what it was. We saw the pH matters, and in fact that most of the variables that matter the most were RIN, pH, and brain weight, of which I didn't discuss, it's a long story, autolysis and worm time, while PMI really didn't seem to matter that much. So one suggestion that could be used for single nucleus RNA-seq experiment is actually to do a light experiment in one brain region, and that may really give you the best predictor of performance on other brain regions as well. So I would like to tell you a little bit about a recent study that we did, a really exciting one for us, but I'm using it also as an example of work that we have been doing with human postmortem brain. So this is work again in collaboration with Steve. The first author is a very, very, very talented postdoc in Steve's lab, Amy Ling, and many others, you can see, contributed to this work. And in this work, we looked at the dorsolateral prefrontal cortex, specifically Bronman area 46, that has been shown together with many other areas to be involved in, to be impacted in schizophrenia. As I mentioned earlier, we had 191 brain donors, 97 controls, and 94 schizophrenia cases. We processed the tissue in villages, as I was mentioning earlier, and you are very familiar with this. So we isolated the nuclei as a single sample and performed single nucleus RNA-seq, and then used transcribed SNPs to identify the donor of origin for each cell. So at the bottom, it shows the different nuclei by donor and then assigned to cell type, including glutamatergic and GABAergic. And Steve and his lab developed a very sophisticated and insightful, so not many surprises there, approach to analyzing this data, which was using machine learning and applying latent factor analysis to bring up the underlying states, the constellation of factors that would justify the many gene expression changes that we were seeing in these donors. So you can see thousands and thousands of differentially expressed genes, but how do you put them together in something that relates to, for instance, cell function? And the beauty of this approach is sorry, is that it allows you to do it across different cell types, so that you not only can see within a particular cell type what genes seem to, what is underlying the overall gene expression, but you can see how different cell types are talking together and changing together. So Steve and his group identified at least 10 different latent factors. 3 of them showed a really powerful relationship between neurons, both glutamatergic and GABAergic, and astrocytes. And we chose latent factor 4 for our analysis because, and I will show you, is affected in schizophrenia. But the fact that at least 3 of these show such a powerful interaction between cells is telling us something that we really should pay a lot of attention, and these analyses are really important as an innovation for these studies, which is the brain function arise from non-cell autonomous processes. Cells collaborate together in order to contribute to specific functions. And the one that is part of latent factor 4 is a collaboration between neurons and astrocytes, which collaborate closely in supporting synaptic functions. So you can see on this graph, on the y-axis is neuronal expression of genes related to synapses. On the x-axis is astrocyte expression of synapse supporting genes. And you can see that they are not only very tightly correlated, and so another important aspect of this study that I should have mentioned is we leveraged inter-individual differences in order to see these changes. Instead of focusing on having a cohort that is as much as possible homogeneous, we really leverage the fact that each individual had its own differences in order to get to these functions and these correlations. So because this is an interaction between astrocyte and neurons affecting the synapse, and you might remember that astrocytes really tightly wrap around most of synapses, around the pre and postsynaptic elements of a synapse. We call this synaptic neuron astrocyte program, so SNAP. And SNAP, as you can see in that graph as well, is decreased in schizophrenia, which was a really, really important finding for us. So I'm going to show you just a little bit more. So these graphs show correlations across glutamatergic neurons and astrocyte, glutamatergic neurons for transsynaptic signaling, and astrocyte for cholesterol biosynthesis, a really interesting finding. Neurons can actually make a little bit of their cholesterol, and it's important to build new synapses, but they actually rely on astrocytes in order to provide the cholesterol to make new synapses. So it's really interesting to see this tight correlation, as well as for synaptic cell adhesions and neurotransmitter reuptake transporters. Interestingly enough, the expression of these factors of SNAP also declines with age. So not only is lower in schizophrenia, but also decline with age in both unaffected control and people with schizophrenia. This is particularly interesting because it is thought that major psychosis or major psychiatric disease represent a vulnerability for dementia. So you could think that if SNAP is already decreased in people with schizophrenia, they start aging at a disadvantage because they're already, their SNAP program is quite a bit deteriorated. And the last thing I would like to share, which was also incredibly exciting for this study, is that when we look at genetic risk for schizophrenia, we tend to focus on neurons. It seems like most of the genetic risk, most of the gene vulnerabilities seem to be concentrated in neurons. But this is because we tend to focus on cell type, not on cell functions, and particularly not on cell programs coordinating between different types of cells. Once we look at SNAP, again, a coordination between astrocyte and neurons, each with different gene families, lowered at the same time, we see that not only SNAP is very much enriched in risk genes for schizophrenia, but they are actually in the astrocyte component of SNAP even more than neurons. So very briefly, what we found, we used inter-individual variation, commonly taught as a nuisance when we receive tissue requests. People always ask to coordinate our cohort in a way that is absolutely identical. That is a potential way to design a study, but you can leverage inter-individual differences to your advantage. We show a synaptic neuron and astrocyte program, SNAP, with gene expression that supports function, synaptic function in a very tightly coordinated manner, which varies among healthy individuals with age, but is also decreased in schizophrenia. And again, it's an important way to look at the impact of genetic vulnerabilities by, again, not focusing on cell types, but focusing on cell programs. I'm going to switch gear a little bit here because when Steve and I started working, our group started collaborating, it was over 7 years ago. And we actually, at the beginning, didn't even know whether drop-seq or single nucleus RNA-seq would even work in human tissue. It turns out it does, and in fact, we can use even tissue that we collected several decades ago for this use. But we did find that there were some parts of our protocols that could be tweaked to make it work even better and to improve the performance of the tissue. And this is something that is really interesting to think about for tissue repositories, because on one end, we need to pay attention to consistency. We need to treat the tissue consistently across the years in the same way. On the other hand, we have to have a crystal ball and try to figure out, will what we are doing today be good for studies that will be done 10 years from now? And this is because it takes for a long time to put together a cohort so that, in fact, the tissue that we just dissected today will be used for several decades to come. So what can we do to know that what we are doing will be the best thing we can do for studies that maybe for assays that haven't been invented or that people are working on at the moment? So this is an invitation to collaborate with us. If you have ideas, and this is something that, again, we have been doing a lot with Steve's group, with Evan McCosco's group, that we are beginning to do with other labs. But the question is, can we work together in ways that we don't change every brain, what we do, but we do things better? And just to give you some ideas on what are the things we could think about, there are things that relate to fresh dissection protocols. Asterisks mark what we do at the moment, but not necessarily what we had done in the past. So when we dissect a tissue, how do we dissect it? At the moment, and this was actually very recently with starting the collaboration with BICAN, we changed a lot of our protocols exactly for these reasons. But we use a cryoprotectant to cool down the brain and then a slicer to slice a 5 millimeter, evenly abandoning the old von Sadel protocol that was the setting by landmarks. There are various ways to freeze the brain: dry ice, cool ice, and pentane. There are several variations. Chuck and us and other laboratories started experimenting on which of these freezing methods may be suitable for different methods and whether there is a difference. And one that it really is on my mind at the moment is really what we do for fixing, and something that Chuck and Evan and us have been discussing quite a bit and will continue to develop, particularly because there are a lot of the emerging spatial transcriptomic platforms that seem to be based on paraffin-embedded tissue. And yet, paraffin-embedded tissue can mean a million different things. The protocols may vary quite a bit It depends on how much it was fixed and many other, how it was fixed. So in other words, it is important that we think about this together and try to figure out ways in which we can really make a difference now for studies that will be done in the future. How am I doing with time? So I'm going to switch gear one more time and tell you of a completely different topic, but that is still related very much to our work. And what I really feel it is one of the greatest challenge that the field of research in general, neuroscience as well, is, which is representation in research. We are becoming more and more aware as a field of how big this is of a problem this is. Just to give you an example, European descent individuals are less than 16% of the world population, and yet they end up making up 81% of a lot of the large-scale genomic data sets that we normally use. This is data from our tissue repository, and I am not proud of it. We consistently have 98% donors are white. So the first thing that we need to ask is why? What is happening? And there are very obvious reasons that we can think of. For instance, there have been historical abuses in research on underprivileged minorities, ethnic minorities. There are inequalities in access to healthcare. There are cultural, religious, and social factors that we need to understand. There is stigma related to medical conditions that may be playing a role. And there are many, many other reasons that intersect, and it may be specific to different communities. So in order to understand what to do, the first thing we need to do is why. What is happening? We might assume that we know the reasons, but we shouldn't. We really should understand them carefully and investigate them. And we need to face the fact that we are part of the problem. We are a big part of the problem. A wake-up call for me was I was reviewing grants, and I read this paragraph: to ensure robust and unbiased results, we will include only Caucasian for this study. One reason this disease is genetically heterogeneous, we need to keep it as homogeneous as possible, and the genetic studies for this disease are only available for Caucasian, therefore that's what we're gonna do. And this paragraph that at face value seems a perfectly fine design really stopped me in my tracks and made me understand that I couldn't continue doing the work I do without trying to understand what could be done to change this and not be part of the problem. So what is that we can do? And I don't have an answer to this, but I can tell you a little bit of the work that we have been doing, sort of experimenting on things that we are hoping will develop ways to address this problem. And one of the biggest one is we need to build trust. We lost, as a medical field, as a scientific field, the trust, or perhaps we never had it, for good reasons. So how do we build it? How do we make this happen? So these are some things that we have been working on, and these are my partners in crimes, two junior faculties at HMS and McLean Hospital. Dr. Folorunso, Timmy is a junior faculty member. He's also assistant director of outreach and community engagement for the Brain Bank. And Caroline Pallavicino-Maggio, who is a junior investigator at McLean Hospital and has done a huge amount of work on outreach and diversity initiatives. So these are three things that we are trying to develop together. One is, and it's obvious, we need to increase diversity in research and clinical workforce. If our workforce is not diverse, we can't hope that participation in research is also equally diverse. We need to be able to collaborate with different groups out of the normal channels, not within the States or within Europe, as we do most of the time. And importantly, we need to figure out a way to develop models to interact with different communities at the local level in a sustainable manner. If we show up and talk about something and then disappear, we have done nothing. We need to be able to engage and learn reciprocally from each other in a continuous manner. So these are things that are kind of ongoing. The McLean Mental Health Research Summer Program is the brainchild of Timmy. This summer will be the 4th year that we are running it. He designed it in a really admirable way. These are 10 weeks of internship paid for students of underrepresented and disadvantaged backgrounds, in which not only they get to work in labs, but the PIs need to commit to mentor them for a lifetime, not for a summer. We are committing to mentor them for as long as they will have a career in science. And something brilliant that the team has done is actually, he has a program to mentor the mentors, so we know what we are doing. But also they participate in workshops and seminars that develop different parts of their professional skills. They do community outreach with us, and they participate in scientific meetings. So these are just some photos. Whoops. Go back. Of many of these students doing this program, but presenting at different conferences, but also working in labs. And this is many of them participating with us in outreach activities as a way to develop this sort of sense of this needs to be done and we all need to participate. It is important that we develop collaborations outside of our regular channels. So this one is a collaboration that started a few years ago with Dr. Molefe and Dr. Node from University of Cape Town in South Africa. They had a new neuroscience department and they were assigned building a new brain bank. They had no experience, so we started collaborating. And I'm really excited to say that the first brain repository in Africa is going to open in 2024 and the end of this year. And it's happening. It took a lot of work on their part, a lot of efforts, but it's happening. And we are working on others in other parts of Africa, including Uganda and possibly Nigeria. Through Caroline, who has a collaboration with Ponce University in Puerto Rico, we are also working to establish a tissue repository in Puerto Rico for the same, with the same goals. And the last one that I would like to show you is the one that is probably the most challenging to do. How do you develop a local sustainable model for community engagement? We informally call our program All-Inclusive Neuroscience, so all in, although this is not a formal name yet. But the 3 goals that we have are learning, again, what are the concerns, the barriers, the attitudes toward participation in research? How can we learn how to cultivate trust? And of course, it has to be a reciprocal program. So we need to be able to offer resources and education as much as we are hoping to be educated as well. So one of the many things that we have been doing, perhaps the most important one, is a collaboration with Dorothy Sebaka. She is a nurse at McLean Hospital, originally from Uganda, amazing person that founded a foundation called Women of Purpose International. And this is meant to focus particularly on mental health of families immigrating to the US, originally from Uganda, and now it is expanded to the rest of Africa and many other countries. Dorothy had set up a huge organization to do this, and now we have been collaborating for the past 2 years. One of the things that we do together is this program that we do in August that has 3 days, 3 afternoons. And I'm going to show you a little bit what we did this past summer just to give you a sense of how it works. So we collaborated with her and her organization. On day one, we focused on adolescents and young adults, so mental health related to younger people, but at all times, everybody was involved at all ages. It was the topic was that. So we had discussion on generational and cultural challenges that children from families immigrating to the US. So the differences in cultural between, for instance, the parents and the children are often a really important issue and one that bears down on mental health. We had several psychiatrists from McLean. Neisha Did is an advocate for mental health for people of color here locally in Boston and also participated. And Dorothy ran the question and answer. Day two, we focused on adults. So conversely, how do the parents deal with the difficulties that they find with their children? But also we had Jonathan Vorgesgan, who is a psychiatrist at McLean and part of our group, to talk about aging and psychiatric symptoms in aging. And then the fun part was day three, neuroscience research. And here we brought in teaching brains. Here you see Thorsten Klengel, who is part of the Brain Bank and one of our collaborators, Timmy, myself, showing different brains and discussing brain anatomy and brain functions and why we have these brains and different disorders that these brains represent. And here is our lovely Kylie training the next generation of neuroscientists on that day. And of course, everything ended up with really inspiring music from our collaborators. So I'll end here to thank people at my coworkers at the Brain Bank, in my lab, Steve and his lab, with whom we have been really enjoying working together, as well as Thorsten Klengel. And of course, the most important thanks always go to tissue donors and their family and to you, the audience. Thank you.
Hey, we're now going to open it up for Q&A. Does anyone in the room have any questions?
Thank you. Really fantastic talk, Sabina. Very exciting. My question to you is, thinking about the diversification of the repository when it comes to brain samples. I mean, obviously, doing it for equity reasons is right, incredibly valuable. But beyond this, the scientific gain that, as you've demonstrated with yours and Steve's work, that how we can leverage inter-individual variation for scientific discovery. Also being able to capture increasingly diverse human genetic backgrounds, I anticipate would also yield also more and more discovery that we might not be able to anticipate today. But could you think of some instances where having access to ancestrally or genetically diverse samples has really yielded, or postmortem samples specifically?
Yeah, it's absolutely, and thank you for bringing it up because it is a really important part. We know that there are differences in many different brain disorders on the vulnerability to different ethnicities. We know that Alzheimer's, for instance, has stronger vulnerabilities in people of color, for instance. And there are many more, and there are more every day that are coming up. And the more we become aware of it, the more it becomes obvious. It can be done, and it's probably easier to change the tide with genetic studies or with clinical studies. It always, postmortem always trails behind only because, again, it takes so long to build a cohort that, in this case is diverse. But it's a really important thing. And it's really something that we are asked all the time when we do this work. Do you know that? Do you know whether we are more vulnerable, less vulnerable? What do we do about it? How would you find out? So absolutely, there are really important work that needs to be done by leveraging any, ethnical ancestry diversity. We just need to work on it And it's important when we do this work that, we don't go in as a bull in a china shop. We can go in and say, donate your brain or participate to research. All we can do is talk about why this work is important, why we do it, what resources we can offer. So it will take time, but it's absolutely essential that we do it.
Thank you so much.
Thank you for the fascinating talk. One thing that struck me was the gap between the neuropathology and the phenotypic manifestation of FTD. So could you elaborate a little bit more about what has helped to fill the gap? I'm assuming molecular markers, but besides that.
So thank you for bringing it up. But this is, again, something that we have been thinking quite a bit. What I was showing is simply the neuropathology. So, we look carefully at the health records. We make sure that the diagnosis was done by a neurologist, for instance, so that it is as accurate as it could get. And then we look at the classic, plain, non-quantitative neuropathology. And day in and day out, we find differences. I was bringing up FTD, but we see it with any brain disorders. We see it with Parkinson, with Alzheimer, with many, many, many of these. And again, I think the reason is because we tend to think categorically. We tend to think Alzheimer has this pathology, FTD has this pathology, but then we can't integrate with what are the symptoms. If you know that, for instance, Alzheimer, the latest from NIH is this sort of ABC system in which you look at 3 different pathologies, neurofibrillary tangles, beta amyloid plaques, and senile plaques, and you put them all together and you give a probability of cognitive decline based on that pathology. 1, we are only looking at cognition. 2, do we really know? And how do we know that those are not simply the sort of messengers of something else? So I think something that I didn't mention, but we are trying to do between the brain bank and the lab is to develop a more transdiagnostic approach to looking at symptoms. And this means doing work such as the one we are doing with SIV, with looking at single cell gene expression, together with a more dimensional measurement of symptoms. But in order to do that, we need to get to symptoms that are not the historical subjective symptoms, they're more biology-based symptoms. So one example that I often make is, if we think about depression, depression is comorbid in pretty much every brain disorder, right? But depression is something that we say, what is depression? It does, if you think about it in that way, where in the brain are you gonna look? But if you start breaking it down in constructs and subconstructs that are more biologically based, so you could think, well, it's anhedonia. So anhedonia, we might want to think about circuits that are involved in reward, a motivation, goal-directed behavior. Once you start thinking in that way and you can develop tools to measure those constructs, then you start getting closer to really understanding what are the brain changes that correspond to symptoms. In my dreams, we're going to do the other way around, and we're going to use the data from biology to find new constructs, to put different bricks together we normally don't consider together, that are not the subjective symptom we know, but are bricks that are biological functions that are disrupted. So it's something that is really, really interesting. And even in data, the way I was showing, this system could be applied, right? So if you have somebody that came in as a diagnosis of Alzheimer, but turned out to be FTD, which happens a lot, were their symptoms different? How do we measure them instead of simply saying, oh yeah, there was cognitive decline?
Thank you. One more question. So you talked about the brain pH varying from region to region. I'm wondering, is there a gradient along the axis, or maybe oligo-rich regions tend to have like this sort of a pH? Is there a clustering based on cell types, or is it
So
varying along the
pH we
axis?
measure on the cerebellum, so it's not region specific. We wouldn't be able to do that because unfortunately to measure pH, we need to take a big chunk of tissue. But RIN is region specific. And to some extent, I mean, a brain might have overall a pretty high RIN, but different regions will have different RINs, or overall a lower one. We haven't figured out exactly what that depends. So initially, we thought about something very simplistic, like, you know, the body is put on their back, so the blood will pool in the occipital lobe, and there are a lot of RNases that could contribute. Typically, the occipital lobe does have a lower RIN, but other parts of the brain, we can figure out how to predict it. So the best way, and fortunately, in order to measure RIN, you only need a very small amount of tissue. So it can, in fact, be measured from the same block you're working from.
Thanks for the interesting talk. Just a question. I've not probably missed the slide. You spoke about the age of the donors.
Mm-hmm.
Which is the age, the median age of the donors, so the youngest ones, or young people donating brain?
Yes.
How young?
So that is a difficulty one, right? You don't want young people to die, of course. So our average age is actually toward the older age. I think we more or less float around 60, 65-year-old as an average age of donors. But people will pass for accidents or for other reasons. So we do have quite a bit of range. Another one of the tissue repositories, part of the NeuroBioBank, actually has had a collaboration in the past with the Children's Hospital. So they have actually fetal tissue and even very young tissue. We are trying to do the same thing with Boston Children's Hospital for the same reason. But typically, children will, you know, there is a peak, unfortunately, of that during the first year of life. And then hopefully very few will pass. And when they do, they have a serious condition. So it is difficult to do that kind of study in human tissue, but there are some resources that are possible.
Not being used for transplantation. Brain is not considered for organ donors, obviously, but it is included when you do the organ donation protocol, because that could be a solution if you add brain for research as an add-on to your will to donate organs for transplants.
Unfortunately, we tried a million times. We tried, and the NIH tried, other brain banks tried, that putting it together in Europe, driver license as an organ donor at the moment is not possible. But people can register to become brain donors. So typically we register people, and it's not a consent, it's not yet a permission to take their brain at that, but it's a way to tell the families what do they need to do and the fact that they wish to become a brain donor.
Thank you very much.
Steve.
So what are the main things that limit the scale of brain donation, and what would be the most straightforward ways to change that and open the floodgates?
It's an interesting question. It would be interesting to you know, it's something we think about all the time, but it's good to think together. For the most part, it is awareness of how important brain donation is. So there are disorders for which it is obvious to people that suffer from them, to their family, how important a brain donation is. And this is in part because they understand that that disorder is a brain disorder, but also because at times we do quite a bit of work. So our own repository historically has been doing quite a bit of collection for brains from people that suffer from Huntington disease. The first director, Dr. Bird, was a Huntington researcher. We continue that tradition and have tried to leverage it and increase it. But it is also easy for people to understand that Huntington is a brain disorder. It's important. And it's really touching that families and even people that suffer from brain disorder will want to donate their brain to help others. It doesn't help them, of course. It doesn't even help their families, but they want to do it. For many other brain disorders, it's much more difficult to raise that awareness. Psychiatric disorders are a good example. We still, as a lay audience, think of particularly some psychiatric disorders as mental disorder, as if we had a mind that is not the brain. So is it the mind or is it the brain that has changes? And therefore, in part because of the stigma, in part because of the lack of awareness of why would we look at the brain of somebody with a psychiatric disorder, we struggle to get brain donations. Even harder it is for people that don't have brain disorders. If you had never heard about neuroscience or have never heard of research, why would you think that donating the brain of somebody that doesn't have brain disorders is helpful? If I had a penny for every time I heard from somebody, oh, I would love to become a brain donor, but I don't have any brain disorder. You can understand why that is a misunderstanding. And the problem is that, you can talk with audiences that are related to brain disorders, advocacy groups or foundations. We do that all the time. We go talk with different foundations. We explain how it is done, why it is done. But there is no foundation of people that don't have brain disorders. So how do you talk to them? So we have been trying to do different things. We collaborate, for instance, with several funeral homes, because funeral home directors are often very good at referring potential donors. The same thing with nurses, social workers. We've been trying to work with religious organizations. That's something that could potentially be a good idea, because these are people that devoted their life to service and may see brain donation, and in fact they do as a service, an ultimate act of service. But it has been a challenge, and a while ago we tried to increase our collection of brains from people that had PTSD, and that turned out to be a really challenging thing, in part because a lot of people that do suffer from PTSD, oftentimes soldiers, don't like the idea that, to be told you have a brain disorder. They are told to be toughened up, to, you're burned out, it will go away, there is no change in your brain, why would you become a brain donor? And, something we try to say is, our brain changes all the time. If you will remember anything that happened this afternoon here, is because your brain changed a little bit. So we shouldn't be afraid of the idea of brain changes. It's only when they become an obstacle to our life that we consider them disorders. But they are so stigmatized that sometimes it's difficult to convey that message. So a lot of the work we do that I was describing, and much more with Foundation, is really focused on try to raise awareness of why it is important to consider becoming a brain donor. Thank you, that's a great question.
Great answer. Thank you very much.
And we do have one question from an online viewer. This viewer says, thank you for the great talk. I wonder if the quality of the snRNA was substantially less good than non-postmortem data.
So actually, Steve can talk better than I can. If you think about other species that can be sacrificed and the postmortem time is relatively low, probably yes, although not to an extent that it affect our studies. We are obviously able to see very subtle differences and very important differences. There has been this idea that we should probably be using, for instance, human tissue from biopsies. And of course, that is a very, very helpful thing to do. It also has its limitations, like anything else that we do. There is a trauma of actually taking the tissue out that is actually degrading the tissue quite a bit, probably more than postmortem that is left as a whole brain, but also the obvious limitations of, you can't study every disorder and have a big cohort with that kind of tissue. So yes, if you are thinking about animal models in which you control completely every parameters, probably there is a difference in tissue performance, but that doesn't mean that this doesn't work. It works just fine.
Okay, well, thank you for a wonderful talk, Sabina.